Okay, so I'm going to take you through a tour of our unit and what will be on your next unit test. So we're starting with Unit 4. You had a review of organelles that needed to be done at the beginning of this semester, or quarter, I should say. And so you want to make certain that you've attacked these really well and that you are super comfortable with those 6 organelles. But don't forget, you do need to know what all cells have. You need to know what structures plant and animal cells have when compared to each other and when compared to bacterial cells. Make sure that you know what a bacteria does and doesn't have. You don't have to know the name of their parts. You do have to know that they don't have any organelles. And how do they keep their DNA? Where is it? And then, as we go through this, we'll just kind of quickly flip through. This was your 2-pager on organelles that hopefully is significantly more complete than mine. And then we started our DNA unit. We added a little bit more information up on the top of this page. This was our small sheet. If you need more details or you want to see these, you can see it a little bit more clearly in your electronic notebook set of slides instead of this video. Then we had our POGL, where we talked about the structure of DNA and how DNA is anti-parallel as well as complementary, and we made certain that we knew our nitrogen base pairing rules. That was a big deal. We then did our DNA extraction lab, where you are going to mount this down on 59, make certain that you had your 3 drawings of our procedural steps, and being sure that you knew why we did each of these things. And on 58, you wanted to draw me a quick little a quick little image of the test tube when you were done with your equipment. If you were not here for that lab, or you were not quick enough to draw the images, if you go to our electronic notebook and go to the page where you'll see this, there's actually a little clickable link so you can see the DNA from the strawberries, the thymus gland, and the banana. Let's keep going. Next, so that was a hinge mount. Next, we went into some models of DNA. I would like very much for you to try this out if you have time this weekend. Otherwise, we'll do it at the beginning of class on Monday, where it's asking you to compare these models of DNA. What's really great about this model versus what's really great about this one, and how about this one? So, for example, I'm showing you that I thought that first model was wonderful to show the double helix shape. And that you can also see that there's 2 different colored nitrogen bases making each of those rungs. You should add more to this if you can think of something else, as well as tell me about these 2 models. What's so great about them? Next, what I've highlighted here is not what you need to highlight. However, when you're reviewing for your test, highlighting is an excellent tool. So make certain that you put really good solid answers here, reread them, make them even better. Check out the highlighting, or you can do some highlighting on your own. We also had page 61B, which is the growth and development of organisms. Mitosis is the only way we can go from a 1-celled organism to a trillion-celled organism. So it's pretty important. So make certain that you review that. Then we went to some very specific details about how we get more cells, and that's the cell cycle with mitosis. I've tried to colorize this here as I was reading. I saw that there was 3 major pieces, and I've got those color marked. There was also a flip page that told me about how the cell cycle and mitosis allows us to grow, to repair our damaged or old cells, and for some organisms they can even reproduce through cell cycle and mitosis. Not sexually reproducing organisms, organisms that have male and female versions of themselves don't do this piece of reproduction. But that was our 2-pager there, and then we had our beautiful cell cycle model. So I tried to keep the coloring the same. I had these 3 colors here, so I went back through my notes and made certain my colors all matched. So whatever is orange here is orange here and orange here. It doesn't matter about your colors, just work through and make certain that whatever you do makes sense to you. Next we got to our DNA replication. I went through and highlighted the most important things, and then you had these 5 questions, and then you answer them back here. The last question, so what happens if the enzymes don't do their jobs? That's really, really important. Don't just tell me DNA couldn't replicate, but tell me the major impact. If DNA can't replicate because the enzymes are broken, then what would happen to the cells? What would happen to new cells? Could we make new cells? Does that mean that we could grow and develop or repair broken pieces? What are the major implications of an enzyme being denatured that normally would help with DNA getting copied? And then on Friday, some of our classes were able to draw this out. This was the last thing we did, and a few of the classes were not able to get this image completed. So please do take a little bit of time and make sure you add those details. You have this original piece of DNA. Helicase splits it, so you can see blue on this side and blue on this side. These are the original pieces of DNA. Then polymerase comes through and adds new nucleotides this way and new nucleotides this way and does some proofreading. And then you'll end up twisting back up and you'll have 2 pieces of DNA that looked exactly like the original, but each of these is half new and half original. So we call that semi-conservative replication. And then we added our chromosome page. This is just a reminder about what a chromosome is, and that the only time we really get to see them is at the beginning of our cell cycle, after interphase has happened, maybe even prophase and metaphase have occurred, so you get those big fluffy chromosomes that have replicated lining up in the midline. We then did our I Know and What I'm Learning. We used 2 different sources. On Monday the 19th, we used a video that was posted on the agenda, and then on Thursday the 21st, we used another video that was posted on the agenda. Make sure that you've got things that you know and things that you're still learning about on both of these pieces. Lastly, were karyotypes. Went through and added a little bit of highlighting detail here, made certain that I had my autosomes and my sex chromosomes labeled, and this was a hinge mount, so we wanted to make sure that we have our references for some of our disorders here. I did go ahead and add some of the words that we were using in class. We used a lot of these words. Some of you know this information and some of you don't. So I'm jotting this down just as a little reminder of some review concepts or some concepts for the first time that some of you are unfamiliar with. Then we went through and we built our own karyotype, and I'm not going to glue and paste all of these down because I was busy helping you glue and paste all of these down. But we want to make certain that you've got your patient A through G, and that you write down what a karyotype is from the other side. Your first 22 pairs are going to be up here, and then you're going to tuck your 23rd pair, which is the sex chromosomes, over there in the bottom box. Do be sure that you label these pieces. Then we had another set of examples with, it looks like 6 more examples of karyotypes, and we did these together in class. And then you had to do 6 on your own. I did the first one together with each of you in class, and then you had 5 more to try on your own. And if I'm not mistaken, the last thing that we did was I handed you a piece of DNA. I'd like you to glue this down or tape this DNA down on page 72, and then I want you to write the complement. Pretend that this is a strip, just the left side of a DNA molecule. If the DNA molecule has a nitrogen base T, then across from it would be bonded an A. If the nitrogen base was an A, then across would be a T, and then a C would attach to a G, and a G would attach to a C. So basically, you're writing the complementary side of DNA down for me. We want to make certain that you also drop a line every 3 nitrogen bases, because that's actually how we use DNA. It's used 3 nitrogen bases or 3 nucleotides at a time. DNA is used to make RNA, and RNA is going to make proteins. So the last thing that I'd like you to write down in your notebook is this little ditty. I want you to remember that DNA is universal, that all DNA is exactly the same. So when we study DNA, we often will just write the A's and T's, C's and G's. All right, I hope your notebook is looking beautiful. Add a little bit of color, go through some of the details, and I look forward to seeing what you know on Monday.